Okay, we are going to talk today about protein synthesis, and I am going to apologize in advance because I am going to attempt to toggle back and forth between my laptop and my iPad to show you some things. So hopefully you can be okay with this. This is brand new to me, so we're going to try it. Okay, so we are going to talk the first part of protein synthesis today, which is transcription. And so when we talk about protein synthesis, we're talking about transcription, which is making DNA into RNA, which we'll talk about in a second, and then translating that RNA template to make a protein. Okay, so a couple of things that you need to understand first, some terms you need to know. The first is called a codon. When we talk about codons, we talk about series of 3 nucleotide bases. It's called a triplet code, but the specific name is a codon. So each codon codes for a specific amino acid. So if we look at the RNA sequence UACCGU, we would look at that as UAC coding for one amino acid and CGU coding for another amino acid. Okay, the genetic code is universal, which means codons code for the same amino acid regardless of species. So if you looked at the genetic code of, let's say, an earthworm and what amino acids it was coding for, they would be coding for the same amino acids that are in us. The sequence would be different. There are 64 possible combinations of those bases, but only 20 amino acids. So as you will see, some amino acids are coded for by more than one codon. So this would be showing you here the codon table. You can also look at codon wheels. It's the same general idea. And what you're seeing here is the idea that UUU and UUC codes for the same amino acids. We're not going to ask you to memorize these, and we're not going to ask you to know what these stand for. Just know that this amino acid here, PHE, has the code UUU and UUC associated with it. So here you can see that SER has 4 different codes associated with it. So when we start talking about mistakes down the road, when we start talking about what happens when a letter is wrong in the code, you could see how, depending on what amino acid was supposed to code for, you may actually end up coding for the same amino acid. So if you had UCU, that was what's supposed to be the code, and you got UCC, you would actually end up making the same amino acid, which is why there are lots of errors that happen that we never know about because the code is the same. Or sorry, the code codes for the same amino acid. If you look here, the next slide's talking about the stop. So UAA, UAG, and UGA are the stop codons, which means basically this is where we stop making the protein. So when we come upon this code right here, we can then stop making the protein. We know we're done. And then this one right here, MET, which has the code AUG. So this is an amino acid, but this code AUG, when we find that code, that's where we start making the protein. So this is going to be where we start making the protein, and any one of these 3 will be where we stop making the protein. And that's what we're talking about here. All right, so we're going to talk through this picture here, back here. So there are 3 stages to transcription: initiation, elongation, and termination. Just based on those words, you could probably figure out to some extent that transcription initiation, that stage, is where it starts. Elongation is where the protein is, the RNA transcript gets longer. And termination is where the transcript stops. So what's going to happen? So the RNA polymerase is going to bind to the promoter, and that is going to start the transcription process. This polymerase here is going to actually add the nucleotides to, it's an enzyme, so it's going to, anytime you see ASE, by the way, it means enzyme. It's going to add the bases to the growing strand. And that's what's happening here. So it bound to the promoter here, and then we started adding in RNA bases to make the protein longer during elongation. So this promoter here is going to decide which side of the DNA we're going to use to transcribe into RNA. So we may use this side to transcribe, or we may use this side to transcribe, but that's going to be decided when we bind here. So during elongation, the RNA strand is going to elongate. It's going to add the nucleotides one by one onto the growing RNA transcript. What's going to happen is the DNA is going to coil back as we move down the RNA. It's going to coil back around into that double helix. So we're basically opening up the double helix, transcribing one side of the DNA into RNA. And as soon as we move past that section, the DNA is going to twist back into the double helix. And this is sort of what it looks like, is that we are getting longer. Here's the RNA transcript. Okay, when we when we finish it, when we hit the termination part, then this will be released, and we will have an RNA transcript that will then go on to the ribosome. So termination is the final stage. The polymerase is going to reach the part of the DNA that is the terminator, which is the end of the gene. And then we're going to release the RNA, and then it can travel down to the ribosome. So let's go ahead and watch this.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides.
So this is the RNA nucleotides that are going to be coming in and matching up with this DNA strand.
DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
Okay, so
oh, hello there. I'm at the gym. I don't know why you're here, but I'm going to do some push-ups.
There we go. All right, well, that was fun. Okay, so messenger RNA is going to be bringing the message from the DNA in the nucleus to the ribosome. So that's the go-between. So the DNA makes messenger RNA. The messenger RNA is modified, and then it leaves the nucleus to go to the ribosome. So we're going to talk about the modification piece next. So the first thing we're going to do is we're going to add nucleotides to the end of the RNA transcript. At one, let's see if I can move this up here. At one end, we're going to have a G cap, and at the other end, we'll have a poly A tail. So basically what those do are those allow the RNA to go into the cytoplasm, and if there were enzymes that came and tried to attack the RNA transcript, it would just take off non-coding parts of the RNA. So if you can imagine this really long piece of information that you really, really, really need to keep intact because it's coding for proteins. So if we add some DNA on one end and DNA on the other end that we don't need, that we don't want to actually be part of the protein, if the enzyme attacks that, then we don't have to worry about some of our code being lost and therefore our protein not being made properly. So it's actually a really cool way of protecting that RNA transcript to get to the ribosome intact. And because we have that start codon built in, we know exactly where to start making our protein. So we don't have to worry about that poly A tail or GCAP being translated into part of the protein, because it's not where the start codon is. So the start codon exists partway into the transcript. So that way we make sure that we don't code anything that we don't want to code for. Another thing that actually happens is, and you may have heard this before, but not all of your DNA is actually used for coding. Some of it is just is there that we don't code for, and when we make the RNA, that non-coding DNA actually gets incorporated into the RNA transcript. So one of the things we need to do is we need to splice the RNA to make sure that we only use coding regions. So the coding regions are called exons, and the non-coding regions are called introns. So what we do is we take out the introns and we splice together the exons so that we only have coding RNA to go to the protein, to go to the ribosome to make the protein. So when it leaves the nucleus, remember, it's got the cap and the poly A tail, and it only has coding RNA, which means that we now have a perfect transcript that will not get damaged in its travels to the ribosome, but also only has coding information. We can watch this again. Hopefully I remember to turn it off afterwards.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process.
So this strand here is the RNA, and this is the DNA, in case you're confused. So the RNA polymerase is moving along the DNA and spitting out the RNA.
Process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons.
So we want to get rid of the green and only keep the yellow.
RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, so you do not need to know the process by which this happens. We just want you to be able to see it so that you hopefully can understand what it looks like. This is a much more simplified version of it here. So if this is an intron, this is an intron, we don't want introns, we only want the exons. So we're going to remove these introns and we're going to splice these together to get our actual our modified RNA. So we're going to stop translation there, and I'm going to attempt to use my iPad to just walk you through transcription. All right, so basically what we have is we have this situation where you have DNA in the nucleus. So let's just come up with a couple of codons. Now, if you see here, TAC, that's one codon, GGG, that's another. So we'll end up with 3 RNA codons. So what happens next is that we will make the messenger RNA. Okay, so remember, in RNA, we do not have thymine, the T, so we're going to have a U. Okay, so we've made our messenger RNA. All right, we're going to assume this is an intron and these are exons. Now remember, this is a whole gene, so this would be much, much, much longer. So we'll end up removing those and splicing this together. Like that. Oops. So we have our intron that was spliced out. We're going to add our GCAP and our poly A tail. And this is our final mRNA that can now leave the nucleus. So once again, we started with our DNA. Let me label that for you. Okay, the DNA opened up. We decided to transcribe this side of it. So there would have been another side up here. Like that. But we decided to transcribe this side here. We got this pre-mRNA transcript. We took out the intron, spliced together the exon that was here and here. We added the GCAP and the poly A tail, and we now have our official mRNA that can leave the nucleus. So that, in a nutshell, is transcription, and next time we will go through translation. So once again, reach out if you have questions, and we'll talk again soon.